Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Raval from Dr. APJ Abdul Kalam Government College, Silvassa, UT of Dadra and Nagar Haveli. Today, we are going to discuss about regulation of glycogen synthesis under the paper Carbohydrate Metabolism. So students, let us see what we are going to learn in this module. We already learned about the glycogen synthesis. We already learned about the glycogenolysis. Over here, our target is to regulation, to find out the checkpoints within the glycogen synthesis. So first of all, we will try to explore the covalent modification of glycogen synthesis. Then we will switch on to the allosteric control of glycogen synthesis, and then last we will discuss the role of hormones in the control of glycogen synthesis. Certain tissues such as the brain and red blood cells rely on glucose for fuel. Serum glucose level must be maintained at about 5 millimolar to maintain a healthy life. We discussed many times in the previous lectures that glycogen is stored either in the skeletal muscles or it is there in the liver. But glycogen that is there in the skeletal muscle is available only for the muscle contraction. But the glycogen that is there within the liver, that is there to maintain the level of glucose within the blood. The serum glucose is maintained by dietary sources, the glycogen breakdown, and synthesis from non-carbohydrate precursors via gluconeogenesis. So mainly by controlling the metabolism of glycogen, we generally maintain the level of glucose within the serum. Glycogenolysis carried out to make available more sugar in the blood. But if it is a more sugar is there within the blood, then the glycogenesis, that is synthesis of glycogen, is to be carried out. But just imagine if these two cycles run parallelly, then it results into the futile cycle. And as you know that the futile cycle results into the loss of energy without in the form of heat, and we do not get anything. Today, I will be briefing you about glycogen synthesis and its regulation mechanism. Glycogen is a large polymer of glucose which residues linked by alpha-1 for glycosidic bond. It has branches at every 10 residue, and these branches are connected with the help of alpha-1,6 glycosidic bonds. Glycogen provides an important energy reserve for the body. As I told you that the glycogen is a source of glucose as and when we require. Glucose is the easily metabolizable carbohydrate. Glucose enters into the glycolysis and then pyruvate is entering into the tissue, and from this we are getting the energy. So it is very important to control the glycogen metabolism. The two main storage sites are the liver and the skeletal muscles, where the glycogen is stored as granules in the cytosol. The granules contain not only the glycogen, but also the enzymes and the regulatory proteins that are required for the glycogen degradation and synthesis. So within these granules, all the enzymes that is required for the metabolism as well as for the regulation is there in the granule. Glycogen metabolism is important because it enables us the blood glucose level to be maintained between the meals via as I told you that glycogen stores in the liver, and it also provides an energy reserve for muscular activity. The maintenance of a blood glucose is essential in order to supply tissue with an easily metabolizable energy sources, particularly for the brain, which uses only glucose except after a long starvation period. If glycogen synthesis and glycogen degradation were allowed to occur simultaneously, it results into the net effect of hydrolysis of UTP, which we call a futile cycle. To avoid this, both pathways needs to be tightly controlled. If we do not control this, we lose the energy in the form of heat and we will not gain anything. So, this control is carried out via allosteric regulation and covalent modification of both the glycogen synthase and phosphorylase. So, what is the concept map for the today's lecture? In this lecture, we will first study briefly about the glycogen synthesis. We will then study the regulation process of glycogenesis, that is nothing but the glycogen synthesis. Lastly, we will explore the biochemistry machinery that controls the glycogenesis. Glycogen is highly branched large polymer of glucose molecule which is linked along its main line by alpha 1,4 glycosidic linkage. Branches arise by alpha 1,6 glycosidic bonds at about every 10th residue. In vertebrates and many microorganisms, surplus glucose is converted to glycogen. Many microorganisms accumulate carbon and energy reserves to manage with the starvation condition provisionally present in the surroundings. The biosynthesis of glycogen is main strategy for such metabolic storage. Glycogen is major intracellular reserve polymer. Glycogen consists of alpha 1,4 linked glucose subunit with alpha 1,6 linked glucose at the branching points. Why regulation is important? If we do not regulate the metabolic processes, then it leads to the futile cycle. Simultaneous synthesis and breakdown of glycogen results into hydrolysis of UTP glucose. It results into futile cycle. Due to this reason, it is very important to control this pathway. Question arises must in your mind, what is a futile cycle? A futile cycle, which is also known as substrate cycle, occurs when two metabolic pathways run simultaneously in opposite directions and have no overall effect other than to dissipate energy in the form of heat. So, if we will not regulate this cycle, then we will lose the energy in the form of heat. The cell will not get anything, but the loss of heat will occur. Now, let's review the glycogenesis process. As you are aware, with the glycogenesis process, the glucose is converted into glucose 6-phosphate by the enzyme hexokinase. This glucose 6-phosphate to glucose 1-phosphate, glucose 1-phosphate to UDP glucose, and that UDP glucose is added ultimately into the growing chain of glycogen with the help of the enzyme glycogen synthase. Glycogenesis and glycogenolysis occur by different pathways. Glycogen synthesis involves the use of an activated form of glucose. Mammals, fungi, and eukaryotic heterotrophic microorganisms use UDP glucose, while bacteria and photosynthetic eukaryotes use ADP glucose. Initially, glucose is phosphorylated to glucose 6-phosphate. This reaction is catalyzed by hexokinase within the muscle or glucokinase in the liver. Phosphoglucomutase catalyzes the isomerization of glucose 6-phosphate to glucose 1-phosphate. Now, UDP glucose phosphorylases catalyze the formation of UDP glucose from UTP and glucose 1-phosphate. Glycogen synthase transfers the glucosyl residue from UDP glucose to the non-reducing terminal of glycogen. It is transferred to hydroxyl terminal of C4 end of the glycogen to form an alpha-1,4 glycosidic bond. Glycogen synthase catalyzes only alpha-1,4 glycosidic bonds. It results into the formation of alpha-amylose. Branching is catalyzed by separate enzyme called branching enzyme. It is also known as alpha-1,4 to 1,6 transglycosylase. Students may refer module 25, that is of glycogenesis, for further detail. So now let's focus on the mechanism of regulation. The regulation is important to avoid a futile cycle. So that's why glycogenesis and glycogenolysis, both pathways, needs to be tightly controlled. This control is carried out via allosteric regulation and covalent modification of both the glycogen synthase and phosphorylase. In addition, the covalent modification is under close hormonal control. We will understand this different mechanism under different titles. That is, we will explore the regulation of glycogen synthesis by studying the covalent modification of glycogen synthase, allosteric control of glycogen synthase. It also involves the hormonal control, that is inhibition of glycogen synthesis by adrenaline and glucagon, while the stimulation of the glycogen synthesis by insulin. Inactive glycogen synthase to active glycogen synthase. There is a difference with the phosphorylated and dephosphorylated form. Glycogen synthase enzyme exists in two forms. They are glycogen synthase A and glycogen synthase B. Glycogen synthase A is an active form of enzyme, while glycogen synthase B is normally inactive form of an enzyme. Glycogen synthase B is converted to glycogen synthase A by the process of dephosphorylation. It is catalyzed by protein phosphatase. Active glycogen synthase, that is a dephosphorylated glycogen synthase, is converted back into inactive phosphorylated glycogen synthase by the process of phosphorylation. It is catalyzed by enzyme protein kinase A. So protein kinase A converts active form of enzyme into inactive form of enzyme. Allosteric control of glycogen synthase. High concentration of glucose 6-phosphate activates glycogen synthase B. Glucose 6-phosphate concentration is low during muscle contraction. Therefore, activity of glycogen synthase B is inhibited. During muscular contraction, phosphorylase B is more active. You refer glycogen breakdown for further details. Therefore, during muscular exercise, glycogen degradation promoted while the glycogen synthesis is inhibited. This is quite obvious. This is important for preventing futile cycle. ATP and glucose 6-phosphate concentration is high during resting stage. This condition inhibits activity of phosphorylase B, whereas glycogen synthase is activated to restore the glycogen. Glucose 6-phosphate does not affect glycogen synthase A. Therefore, glycogen synthase A form is active and does not affected by the concentration of glucose 6-phosphate. In liver, phosphorylase B is not activated by AMP, that is adenosine monophosphate, and therefore always inactive. So glycogen degradation in liver is not responsive to the energy status of the cell. Rather, phosphorylase A is deactivated by glucose. This fits with the different role of glycogen storage in the liver, namely to maintain blood level of glucose. Thus, as glucose level rises, glycogen degradation by the liver phosphorylase A is set off, and degradation starts again only as the glucose level falls. This is quite obvious. If the glucose is not available, obviously glycogenolysis is promoted, and if glucose is available, then glycogenesis is promoted. The anabolic pathway contrasting with the glycogen breakdown is that of glycogen synthesis. Just as a cell reciprocally regulates glycolysis, gluconeogenesis to prevent a futile cycle, so too do cells use reciprocal schemes to regulate glycogen breakdown and synthesis. Now let's try to review the hormonal control, how glycogen synthesis is controlled with the help of hormones. First, we will study the inhibition of glycogen synthesis by adrenaline. Glycogen synthesis and glycogen lysis is regulated by hormones. When level of blood glucose falls, alpha cells of pancreas secrete the glucagon. Glucagon stimulate glycogen lysis inside the liver. Glycogenolysis releases the glucose into the bloodstream to improve blood glucose level again. Flight or fight response stimulate the adrenal medulla to release the adrenaline. Adrenaline binds to the beta adrenergic receptors on the plasma membrane of the target cell. It causes a conformational change in the protein. It activates the G protein, which in turn activates the adenylate cyclase enzyme. Activated adenylate cyclase converts ATP to cyclic AMP. The cyclic AMP binds to the cyclic AMP dependent protein kinase. The active protein kinase A phosphorylates the phosphorylase kinase. Phosphorylated phosphorylase kinase is active form of a phosphorylase kinase. It phosphorylates serine residue in phosphorylase B, which converts it into phosphorylase A, that is a more active form. This promotes the glycogenolysis. Active glycogen synthase, that is dephosphorylated glycogen synthase, is converted back into inactive phosphorylated glycogen synthase by the process of phosphorylation. It is catalyzed by the enzyme protein kinase A. Hormonal control again, but Over here we will study the inactivation of glycogen breakdown by insulin. The effect of insulin is opposite to the effect of glucagon and epinephrine. Insulin binds to a receptor on a cell surface and activates a pathway that directs to activation of protein phosphatase 1. Insulin is released in the blood by pancreas when blood glucose level is high. Insulin binds to the receptor on plasma membrane. Binding results into the activation of the receptor. Activated receptor has a tyrosine kinase activity. It will activate insulin responsive protein kinase. Activated insulin responsive protein kinase phosphorylates protein phosphatase 1. It will activate protein phosphatase 1. Protein phosphatase 1 ensures dephosphorylation of phosphorylase kinase. Dephosphorylation makes it inactive. Dephosphorylase kinase that is inactive is unable to phosphorylate glycogen phosphorylase B. It results into the decrease in the glycogen breakdown. To summarize, a glycogen is a branched polymer of glucose that serve as an osmotically neutral means to store glucose in cells. It is present in organisms from bacteria and archaea to humans. Glycogen degradation and glycogen synthesis are controlled both by allosteric regulation and by hormonal control. So, hormonal control as well as allosteric modification, the covalent modification, is important in the metabolism of glycogen. So, students, let's try to conclude what we have learned in this module. Glycogen is stored mainly by the liver and the skeletal muscles, as we discussed, as an energy reserve. Stored glycogen is within the muscle, is available only for the muscular movement. The role of stored glycogen in muscle is to provide a source of energy upon prolonged muscle contraction. In contrast, glycogen stored in the liver is used to maintain blood glucose levels. So, function of the both stored glycogen is different. Glycogen provides an additional source of glucose besides that produced via gluconeogenesis. Because glycogen contains so many glucoses, it acts like a battery backup for the body, providing a quick source of glucose when needed and providing a place to store excess glucose when glucose concentration in the blood rises. The branching of a glycogen is an important feature of the molecule metabolically as well. Since glycogen is broken down from the ends of the molecule, more and more branches translate to more ends and more glucose that can be released at once. Liver and skeletal muscles are the primary sites in the body where glycogen is found. In this lecture, we learn about the regulation of the glycogen synthesis. We learn about the role of hormones and different enzymes in the regulation of glycogenolysis. Hormones have an influence on the glycogenesis as well as glycogenolysis. Over the last 20 years, improvements have been achieved in understanding how glycogen is regulated at the molecular level and how it is connected with other biological processes. With emerging new technologies, the next decade will be undoubtedly give us a better understanding of features which at present are not completely understood. within the carbohydrate metabolism. There are involvements of the sRNA in regulation, but yet it is not clear. So, in future, we will come out with new developments that will clear the idea about the regulation of a glycogen metabolism. Thank you very much.